too often, but welcome anyway. Rare diseases affect 7% of people at some point in their lives. That's 3.5 million people in the UK. Rare Disease Day is an international day to raise awareness, and 2018 is the 11th year of that day. It's the 8th year that we've celebrated it here at Royal Holloway. It is usually celebrated on the last day of February, so that's obviously a leap. In a leap year, it's a rare day in itself, so we're a bit further forward this year. And we expect that it's going to be celebrated in about 100 countries. The theme for this year in 2018 is research, and research plays a really important part here at Royal Holloway. It leads us to take on new discoveries, and those discoveries themselves translate into different forms of benefit for the people who are the users of those. And research also at Royal Holloway benefits our teaching as well, and the things that we lecture and study here. So it's a very important part of what we do. Gene and cell therapies provide treatments for some rare diseases, and at Royal Holloway this is a real research strength that we have. You're going to hear today a lot more from our gene and cell therapy specialists about the different ways in which their work has addressed some of the challenges of rare diseases. And so just finally, I hope that you do enjoy the day. I know there's some talks this morning and then a lot of activities for you to do later in the day. So thank you for coming here, and do enjoy this very important part of our work. Thank you.
Thank you very much, Katie, for this really kind introduction. Thank you for your patience, those of you who were here on time to start. And as Katie has introduced, we run a Rare Disease Day event. We have done it for 8 years now. This is the theme for the year. And this is what I intend to cover in my presentation today. I'm going to tell you what is a rare disease, why is there a Rare Disease Day, why is this important specifically at Royal Holloway. We'll talk a little bit about genes and rare diseases. We'll discuss whether all mutations in DNA are actually bad, and what progress is being done in this field of rare disease. We'll talk a little bit about marketed products and their pricing, and we'll finish on a note on how inspirational people who are affected by rare diseases are. I'll also give you an idea of what other activities you are going to be doing during the day, and we'll hope that you will enjoy and be inspired by what you are going to see throughout the day. There is actually a formal definition. It varies in different regions of the world, but in Europe, a disease is considered rare if fewer than 1 in 2,000 people are affected by it. But the issue about rare diseases is that there are loads of them. We don't actually have an accurate number. We say there are somewhere between 6,000 and 8,000 rare diseases. And as Katie mentioned, if you take them all together, they affect 7% of people at some stage in their lives. If you calculate your numbers in the UK, that means that 3.5 million people will be affected by a rare disease at some point in their lives. And because we really have no treatments, those diseases take a disproportionate percentage of the health budget. They take about 20% of the health budget. In addition, most rare diseases affect children, and 30% of them will die before their 5th birthday. So they are very severe diseases. And actually, most of us don't know about this. So raising awareness about this is a very important issue. Again, as Katie introduced, this takes place in the last day of February. It's the 11th edition this year, and we at Holloway have run an event for 8 years now. The main objective is to raise awareness among the public, but also among professionals and decision makers. And any stakeholder in this field really is more than welcome to become involved. Why is this of importance specifically at Royal Holloway? Well, About 80% of those rare diseases are inherited, are passed on from parents to children. Many are potentially treatable by genetic and stem cell therapies, and Royal Holloway has an international reputation in this particular research field, and you will be introduced to some of the work we do here in a few minutes. So we do showcase our research and teaching on Rare Disease Day. Just to give you an idea of the people working here in this area in both gene and stem cells, and specifically Professor Dickson will be talking to you in a few minutes about Duchenne muscular dystrophy, one of those rare diseases. Again, just to give you an idea, these are the diseases that we work on. Some of them are rare: spinal muscular atrophy, ataxia telangiectasia, the muscular dystrophies, motor neuron disease. Others are not necessarily rare, but the technology that is being developed to treat these diseases by gene and cell therapy will be applicable to common diseases eventually. In fact, currently it's being applied to leukemia very, very successfully. What I want to do in the next few minutes is just refresh your memory, so to make sure that there is an understanding of what's the molecular basis of everything that we are going to discuss now. So what you are just about seeing here with this slide is what we call a metaphase spread. So those blue blobs are the chromosomes, and a little better you can see those green dots. Those label one particular gene in the human chromosomes. We have about 20,000 genes. And those genes store the information to make proteins. That's what genes do. A normal gene will make a normal protein, but a mutant gene, and the mutation is represented like a green blob here, would perhaps make an abnormal protein or make no protein at all. So how important is that? Well, some mutations are actually irrelevant in the sense that there is redundancy in the system and some genes can cover for each other. So if one is mutated, perhaps nothing matters. In some cases, the effect is minor. You may have a minor issue, but perhaps not necessarily a disease. And evolution is based on mutations, and obviously evolution is critical for us to adapt to changing environments. So mutations have a critical role to play in life. But actually, in some cases, it can be very different, and a single mutation in a gene can cause a very important disease. And I'm specifically mentioning here Duchenne muscular dystrophy and spinal muscular atrophy because you are going to hear about them in the next talks. But just to give you an impression of how important, how common this is, there is actually a formal list of rare diseases. These are only the few at the beginning of that list. So cutting a long story short, I can tell you that if you were to read aloud the list of rare diseases, that would actually take you 12 hours to do. If you consider that each of rare diseases is an incredible amount of work, if you want to, in terms of trying to deliver diagnostics, a therapy, apply standards of care to it, the field is incredibly huge. That's the only word that can describe it, really. It's an incredible challenge. Diagnosis can take a long, long time. Some people wait 5 years. In some cases, there is never a diagnosis because the disease has not been reported before. And as you can read here, because of the complexity of those diseases, in an ideal world, not yet really being applied, this is what you would want if you had a rare disease. So there are patient organizations and lobbying bodies like Rare Disease UK, which is going to exhibit at our exhibition, that do a really important role trying to progress the field in a variety of aspects with the goal of improving care for patients affected. So Is there hope for rare diseases? Well, there is. There are significant successes in gene therapy clinical trials, spinal muscular atrophy, as you are going to hear in a minute, in several immunodeficiencies when the body is unable to fight infections, in hemophilia, and in other diseases. And there is an international rare disease research consortium with declared goals of developing diagnostics for most rare diseases and a cure for 200 of them by 2020. And they have been so successful that they have actually updated their vision for 2017 to 2027, and that is their current goal. So certainly there is progress here. But it's not all about developing a therapy, because for a handful of diseases, actually there is a therapy that has been marketed, has been through all the clinical trials, has been approved by the regulators, and is for sale. But as you can see there, regardless of whether the therapy is an engineered virus like Glybera, or is a genetically modified cell like Strimvelis, or is a relatively small molecule like Spinraza, the prices are quite astonishing. So access to those drugs is a very significant problem in the rare disease field, because as you can see, they are quite unaffordable. So this is something that the field is grappling with at the moment. So what I'm going to do in the last minute or so is give you a flavor of what you are going to be doing for the rest of the day. You are going to be, after our lectures, attending an exhibition of stakeholders in the field that are going to showcase the work they do, lobbying or supporting people affected by rare diseases. You are going to do a lab activity that is going to show you how particular immunodeficiency is inherited. You are going to have a speed dating event with our university undergraduates to discuss a variety of aspects of university life and rare disease. And you are going to do an activity, we call it Disease Detective, in which you are going to try to guess what is the rare disease that affects specific people using fact sheets. And we hope in those, say, 5 activities that you are going to be doing today, that we will be presenting quite a complex picture of what's going on in this field. My last note is on how inspirational people who are affected by rare diseases can be, because what those people try to do is focus on what they can do, not with the things that they cannot do, what they can actually do. So for instance, in past years, our patron for the day has been Helene Rainsford, who is affected by a rare disease of connective tissue, but that didn't prevent her from becoming a gold medal in the Paralympic Games. Or Srin Madipali, who is affected by spinal muscular atrophy, but as you can see, even though he is wheelchair bound, that hasn't stopped him from doing a variety of things. And actually, his company, he is the co-founder of a company, effectively the disabled Airbnb, which has been bought up by Airbnb, and he is now relocated to San Francisco for developing that particular area of Airbnb for disabled people. So these guys are incredibly inspirational, and it's something that we all really should take example from. And obviously, to organize an event like this, a number of people are involved, so we are very thankful to our School of Biological Sciences here, to Research and Enterprise in Holloway, the Student Recruitment and Partnerships, and Communications and External Relations. But overall, we are really thankful to staff, postgraduate and undergraduate students who you will see around today developing all those activities, to our exhibitors who come and invest some of their time showcasing the work they do, and our speakers for today. Those speakers you have on the program will we have had, unfortunately, a change on the program because Vanessa Christie Brown is unwell, but very luckily, James Sleigh has accepted to stand in for her, and he will showcase the work that the Spinal Muscular Atrophy Trust does in this area. Then Professor George Dickson will give you an idea of the research that is going on Duchenne muscular dystrophy. And finally, Christos Kapourani, who is affected by a rare disease of the skin, will give you the very personal picture of what it is life when you are affected by one of these rare diseases. And what we hope at the end of the day is to inspire you, perhaps to in the future become involved in one way or other in this particular genetic revolution that is going on. Thank you very much.
Thank you, Rafael. As Rafael said, my name is James Sleigh, and I'm standing in for Vanessa this morning, who is a member of the charity SMA the SMA Trust. So you have to bear with me with some of her slides. So I'm a scientific researcher, and I work on a number of different rare diseases. And in my spare time, I also work for a charity called Spinal Muscular Atrophy Support UK. And today, Rafael's asked us to talk to you about the role of these different rare disease charities in supporting patients and families, and what their function is. So, using the SMA Trust and SMA Support UK, hopefully I can explain this to you. So, in the next few minutes, I'm going to talk to you about why research on rare diseases is important, and this is essentially the more we know about rare diseases, the better we will be prepared to ultimately find treatments and therapies for these conditions, and hopefully one day come up with cures for them. I'm then going to talk to you about where charities, the rare disease charities come in and their function, and use the SMA Trust and my limited knowledge of the SMA Trust as an example and case study. So, why is research on rare disease important? As I mentioned, the more we understand about a disease, why it occurs, when it occurs, how best to treat it, and what impact it has on patients and families, the better we will be able to find treatments and ultimately cures for the diseases. So research is very important for this, but it's also important to find out how to measure a disease. And that sounds a bit strange, but ultimately, if we are to come up with treatments for these diseases, we need to be able to measure how effective they are. So the more we know about how a disease progresses and how therapeutic intervention can affect this, the better. And then finally, as Rafael mentioned, it often takes a good number of years to be able to diagnose rare diseases because we don't know a great deal about them. So again, the more we know about these rare conditions, the better it will be to identify and diagnose them. So getting from what I do in the laboratory, which is study these different conditions, ultimately to providing a patient care and therapies is a very long-winded and expensive process. So just a quick look at some stats this morning. On average, we're talking at least 20 years to go from the lab all the way to providing a treatment, and we're talking in excess of £2 billion. So it's a very difficult and arduous process, which Vanessa here has split up into three major sections. So we've got discovery, development, and delivery. So I'm very much involved in the discovery element with my work in the laboratory, where we're using various different models to be able to understand how these different rare conditions are occurring, and we're coming up with ideas to basically try and understand the disease so that we can better create and design therapies, the gene therapies and stem cell-based therapies to treat these diseases. Then during development, we have to go through a number of different clinical trials, so starting with smaller phase 1 and progressing through phase 2 and 3 to test and see whether the therapies that have been identified in the laboratory preclinically have a benefit in patients. And then ultimately, once a drug is proven effective, we then have to go through various regulatory approval processes. And in Europe, we need to, the European Medicines Agency needs to approve a therapy for it to then be delivered and be effective and useful for patients. There's a very, very long-winded, difficult process. So where do rare disease charities come into this process, and what role can we play? So in the next couple of slides, I'm going to hopefully answer why or what role does rare disease charities play, what assets they can bring, and ultimately how can we make a difference to patients and families. So the SMA Trust is a charity that supports, as Rafael said, patients with spinal muscular atrophy, which is one rare condition, but we've just heard that there are many different rare conditions, and a lot of the different charities that have been set up to support these different diseases often come from family members affected by a condition that their GPs and their doctors know very little about. And the SMA Trust is an example of this, where two grandparents of young babies with spinal muscular atrophy came together to set up a charity that could support others who were impacted by the disease. And so the SMA Trust was born 10 to 15 years ago because of this. And the role of the SMA Trust and other charities is to principally fundraise and create and raise money to be able to raise awareness and fund research. And so SMA Support UK, which is the partner charity which I'm affiliated with, is very much also involved in supporting patients and families, so providing care packs, providing information to families who have had the diagnoses of spinal muscular atrophy. So the SMA Trust has over 15 years raised £4 million for research, and this money has been invaluable to the research community, but also to the patients. So ultimately, the SMA Trust this money has gone towards funding collaborative research. So laboratories I've worked in before have benefited very much from the funding that the SMA Trust has very kindly raised. They also provide an excellent network of researchers and provide a way for researchers to interact, for instance, at scientific conferences. Importantly, I've benefited also in the past from pump-primed research or seed funding, where if we have an idea in the laboratory to be able to test a few ideas about a therapy that we then want more money in the future for, they'll provide little sums of money for us to test little ideas. And also they provide much needed resources to scientists, as I said, to meetings and funding positions like myself, where I'm explaining and hopefully increasing the understanding of these diseases from a research standpoint. And very importantly, the SMA Support UK, we provide information packs to newly diagnosed families, and we're there as a support network to help them get through and understand the diagnosis of SMA. So to summarize Vanessa's talk with a few key take-home messages, the quest, as I've mentioned, to develop therapies for rare diseases is very lengthy and a very expensive process, and something that requires a number of different people, including scientists, rare disease charities, universities, pharmaceutical companies, all to come together to work in this process to best develop these therapies. So rare diseases, as I said, can make a very big impact on this quest, and rare disease charities are driven by a very strong patient need, because we have very few examples of therapies that we can use to treat rare diseases. And as it says here, often the biggest the experts on these different rare conditions are the researchers, but also the people who run the rare disease charities. They've been working on these diseases for many years and have come to understand the requirements of patients and their families, and have a good understanding of what research is being done. And they're always looking to use clever ways to make the money that they do get, because we're always competing for money from the bigger charities, for instance, British Heart Foundation. Many people give to those charities, but it's very difficult as a rare condition to generate that money. So coming to raise awareness of rare disease is very important. And ultimately, the research that goes on into these diseases, we can often find out a lot about basic biology of humans and the nervous system or whatever condition is being worked on. So it not only has an impact on the disease, but also our basic understanding of human biology. So thank you very much. That was just a kind of a highlight of how rare disease charities can have an important role in understanding diseases. Thank you.
So, good morning, everyone. My name's Professor George Dickson, and I've been given the task of talking to you about a particular inherited disease that we have an interest in at Royal Holloway. And the title of my talk's a little bit provocative, GM humans, because I know this is often an ethical issue that's debated at school, but something you maybe want to take forward in the future and think about this a little bit. Is someone who has had a kidney transplant, for example, or a blood transfusion, a genetically modified human being. And so some of the concerns about gene therapy may be alleviated thinking along those lines. Now, the Human Genome Project is very old now. We have 100 or so human genomes here in the audience, I see. But the impact of that project has been very wide, in particular on understanding the basic mechanisms of diseases, particularly inherited diseases, and in searching for treatments. Now, my interest is in Duchenne muscular dystrophy, named after a famous French neurologist, Guy de Boulogne. His surname was Duchenne. And we've worked for many years searching for treatments to this disease. Now, Duchenne is a so-called X-linked inherited disease. That means that actually the disease manifests only in boys who, in the audience here, have only a single X chromosome, as shown here on the right-hand side, this single X chromosome, as opposed to ladies in the audience who have two X chromosomes. So if that chromosome is damaged, a boy will get the disease, whereas with a lady, if she has a damaged X chromosome, the other chromosome will compensate, and she becomes a carrier for Duchenne muscular dystrophy. Now, this is a disease of skeletal muscle. I'm showing you here 4 human skeletal muscle cells. The striated structures here, again, some of you understand this, is the contractile apparatus of skeletal muscle, the molecular mechanism that muscle actually contracts, and the blue ovoid or circular structures are the nuclei of the muscle cell. Now, the gene that's affected in Duchenne muscular dystrophy on the X chromosome codes for a particular protein, and if we turn these muscle cells so they're in a sense coming out of the screen towards you, they're cylindrical structures, you can see on the right here that those cylindrical structures are being outlined by an antibody, a histological stain, with an antibody to the product of the Duchenne muscular dystrophy gene, protein, I've said, called dystrophin. And you can see it coats the inside surface of the muscle fibers. In Duchenne, the protein is largely missing, and that's shown here on the right, much lower levels. And with that, there becomes a disease pathology. Fibers, these contractile muscle fibers are lost. And if you look at a little more detail in the histology of the muscle, we see that there is damage to the fibers, these red markings here. There's inflammation. All these little dots are inflammatory cells flooding the tissue, and the muscle is gradually replaced by a scarring, a fibrosis. And that pathological process is what causes the progressive muscle wasting and loss of strength that's characteristic of Duchenne muscular dystrophy, beginning at about the age of 2 or 3 in youngsters who before that appeared perfectly healthy. And it's a progressive muscle wasting disease which leads eventually to youngsters being forced to be wheelchair bound or to use wheelchairs, and eventually, I have to say, it's a life-threatening condition when the breathing and cardiac muscles are affected later in life. And well, there are approaches to treat this disease. There are surgical approaches, for example, to maintain the spine in a correct structure, and there are very serious drugs, steroids, that can be used to retard the disease by 12 months or 24 months. Really, there are no genuinely effective treatments or cures, could I say, for this particular disease. Now, if we know the gene and the genetic product, this has led to attempts to provide a genetic therapy for the illness. And there are 3 flavors to these genetic therapies being developed. They're shown here. There's so-called exon skipping, complex technology, antisense therapy, which corrects the genetic machine. There's a gene addition therapy, which adds a functional synthetic gene to replace the damaged one. And there's something called genome surgery, which repairs the actual damaged gene itself. Now, first of all, the exon skipping strategy in Duchenne muscular dystrophy, what does it actually mean? What's involved here? Now, some of you who are studying biology will have come across the concept that genes come in small packages. These packages are called exons. They're eventually brought together to provide the coding sequence for the product of the gene in the messenger RNA. What this technology does, it uses synthetic DNA, small sections of synthetic DNA, to interact with exons that are damaged or have errors in them, in the gene. And instead of them being included in the gene, they are excluded by a process called exon skipping. Now, in the case of Duchenne muscular dystrophy, this type of exon skipping therapy has been under development for many years. And what I'm showing you here is just an example from a clinical trial of this type of drug administered through the bloodstream in patients with Duchenne muscular dystrophy over about a year period. And what you can see on the dark line is that in treated patients, the disease is stabilized. This is a measurement of exercise tolerance. It's called the 6-minute walk test in patients with Duchenne muscular dystrophy. Whereas in an untreated group of patients, unfortunately, their ability to walk and move continues to decline until they are also moved on to the therapy. This is this grey box, where you see again a stabilization of their condition. And actually this medicine is called Exon Dis 51, produced by a company called Sarepta, is still in ongoing clinical trials, but has been authorized by the US FDA, the Food and Drugs Administration, as a medicine which can go into the pharmacy freezers or on the pharmacy shelves and be administered by clinicians to patients with Duchenne muscular dystrophy. Now, there are other forms of this type of medicine being developed. Here at Royal Holloway, we were involved in studying the Exon 51 medicine, but we've also developed other new medicines which are now also deep into clinical trial. And these clinical trials are examining newborn DMD patients who can walk or who are already in a wheelchair. And the treatment here, we hope, may be viable for a significant proportion of patients with Duchenne muscular dystrophy. Secondly, I'll talk about gene addition therapy. So this is where we add a functional synthetic gene. We know what the gene is that's affected in this disease. It's on the X chromosome. It's in these nuclei. It's malfunctioning. Perhaps we could substitute that damaged gene with a healthy version produced and manufactured in the laboratory or in the factory. Now, do we have these lab-based healthy DMD genes? Well, actually, yes, of course we do. As many of you will know, there's been a recombinant DNA revolution, and we can now take and grow and expand human genes in structures called plasmids, which can be grown inside bacteria. So this allows us to manipulate, identify, and characterize healthy versions of the Duchenne muscular dystrophy gene. Second question, well, we have that. Can we then administer them safely into patients or into animal models? And the answer to that is yes, it appears so. To do that efficiently, we need a structure like this. This beautiful organism is an adeno-associated virus, and we can insert the Duchenne gene into this viral structure and then use it to infect, or what's called transduce, patient skeletal muscle and restore expression of the missing dystrophin protein. So, the question, of course, is do they work to alleviate or prevent Duchenne muscular dystrophy? Well, the answer is yes, a tentative yes. But at this point in time, that's been shown only in animal model studies, where the gene therapy treatment is effectively curative or with profound improvements in larger veterinary animal models of muscular dystrophy. Now, where are we with this? How do we transfer these observations into human context? Well, we know that the gene therapy is effective. It works in the long term, improves the clinical status of the patient, and improves survival in these particular models. And it's feasible for indeed all Duchenne muscular dystrophy genotypes. And this encourages us and others to move to human clinical trials, so-called phase 1 early and slightly more advanced phase 2 clinical trials. And these are happening, are being developed in the UK and in France in a collaborative effort. And also across the world, there are a number of other companies and organizations taking this type of Duchenne muscular dystrophy gene therapy forward into clinical trials. And the next few years will tell us whether these approaches are going to be successful or not. So a very exciting time. So that's the second strategy, genetic strategy. The third one is a little bit more experimental, indeed, a little bit more advanced. It's what I call genome surgery, repairing the damaged gene. What does this mean? What can we do in the lab and maybe eventually in the clinic here? Well, this slide is kind of complicated, but it's an example of what might be achieved. These numbers here refer to the blocks of the DMG gene, the exons. It runs away out from here, from number 1 all the way to number 79. But a number of patients have a deletion in the gene of the blocks between 44 and 53, 45 to 52 are missing. And what this new technology involves the development of specialized enzymes which can cut DNA, a bit like a restriction enzyme, if you've come across that in any of your classes. So it can cut the chromosome in a specific location, and actually this is on the X chromosome specifically. And at that cleavage point we can then add a repair template with the missing exon blocks in place and restore a full-length functional gene. Now, can that be achieved? Well, the answer is yes, it can be achieved. Here at Royal Holloway we've shown that in some very early studies, way back 2013. And we were one of the first teams to demonstrate this type of technology could be active. And actually, this type of technology is probably going to be applied specifically to something called a muscle stem cell. I just thought it'd be interesting for you to see a muscle stem cell in action here. So this little movie I'm going to show you is of a muscle fiber. This long structure is the contractile muscle fiber. You can just about see the striations in there. And these structures around about it are the stem cells of the muscle. And if you watch them, you'll see them sort of scurrying up and down the fiber here, dividing from 2 to 4 into larger numbers of cells. And these are actually the stem cell population that repairs the skeletal musculature when it's damaged. And this type of genome surgery technology will probably initially be applied towards those types of stem cells in the skeletal muscular system. So really, to wind up, we're in a phase James described of translational research. I often put this slide up because 1953, we all know who these guys are, of course, Watson and Crick. We knew nothing, or we didn't know about the structure of DNA until they defined it. And over the subsequent 50 or 60 years, we're now in a position where we have therapeutics based on genetic material now in the clinic and moving forward into generalized application for patient benefit. So it's exciting times. There's a case for almost individualized medicines here. It's a difficult regulatory landscape. People are concerned with genetic modification and what are the best pathways for drug approvals and so on. And actually many of us are still working to produce better and more effective versions of these extremely powerful genetic medicines. This has been mentioned. All of this work is performed in partnership. How are these sort of processes achieved? Partnerships, particularly financing this type of research. Patient groups, some of which you will meet today in the exhibition. In my field, it's patient groups like Muscular Dystrophy UK or Action Duchenne. So please go along and chat to those groups who have really pioneered the work leading to these treatments. But then all the way through from hospitals and universities right through to the biotechnology companies that will put these sort of medicines on the pharmacy shelf. So I'm finishing up. As said earlier, there's 100 or so human genomes in the audience. There isn't one human genome, there are many, and slight changes in that genome can produce the rather beautiful and interesting world that we live in. And it's kind of an extreme example of the Great Dane and the pug, who are virtually identical genetically, but very different phenotypically. And of course, some of these changes are beautiful, but some of these changes can be detrimentary and produce illnesses like Duchenne muscular dystrophy. And I just wanted to put this slide up to remind any of you who are thinking of careers in medical research that it's not all heavy-duty stuff at the lab bench, and there is a little bit of fun and enjoyment associated with science. And this just gives you a picture of some of the people in the gene and cell therapy group at Royal Holloway who are having an afternoon off and a picnic down the road in Windsor at Great Park. So science can be fun as well. So thank you, and I guess we'll maybe have questions at the end of the session. I'll stop there.
Yep. Hello everyone, my name is Christos Kapourani. I'm 33 years old and I'm originally from Greece, and I suffer from recessive dystrophic epidermolysis bullosa, which is short for EB. And I was diagnosed just 2 days after I was born, so I was the third sibling. My brother was already with EB when I was born, and we had already lost a sister to the same condition. Growing up with EB has been very difficult because I grew up in a country where there was not a lot and they didn't know much about EB and how to treat it. But I was lucky because my mom was a nurse in her profession, so she knew how to take care of me and my brother. And so every day I have to go through a lot of dressing changes, maybe 2 to 3 hours a day. So the condition is very, very difficult because the skin is so thin, which is a lot of people compare it to a butterfly skin. So it's so sensitive, even the slightest touch, rub, or even a handshake could make damage to the skin. And then that skin, then if it's damaged, it will blister, and then that blister will turn into wounds. So, which is why most of EB patients cover their body with bandages, and that's why those bandages take ages every day to change. But even though I have to go through all these struggles, I have tried to carry on with my life, make friends, go to college in Greece, and I've had a lot of problems. But in the future, I was directed to the London Hospital St. Thomas's from a neighbor of mine in Greece, and they said that they have a hospital there which specializes in epidermolysis bullosa, EB, and in the year 2000, I first started coming to the hospital. I had hand surgery because with EB, the hands, the web spaces, they tend to close and they become like a fist. So that's when I had my first hand release surgery to have a proper hand. And since then, I've kept my hands quite well by using dressings, gloves, as you can see, so that the web spaces don't close. And then every year I used to come and go from Greece until the year 2005, which my condition became worse because I couldn't swallow or eat any solid food. Because EB doesn't affect only the outer skin, it affects the internal organs as well, such as esophagus, kidneys, heart, and all other organs as well. So in 2005, my parents and I, we decided to move to the UK with the help of Deborah to settle here because the government wouldn't let me go back and forth every 3 months that I needed to come over here and have esophageal dilatation so I can have solid foods again. But since then, I think it's been the best decision I've done for myself because coming here, I learned how to drive, first of all, did a foundation degree in architecture, and then from that, I went to Greenwich University, did another degree in 3D design and animation, and then in 2014, I graduated with a 2-1, which I'm very happy because it's my biggest accomplishment I've done so far. But as soon as I started to get myself back from university and relax a bit, in 2015 I was diagnosed with skin cancer, which is the biggest threat to epidermolysis bullosa. But luckily the doctors did the surgery and removed everything, and it's been up and down since then because 6 months later another cancer appeared and then another surgery was needed. So as soon as I was trying to get my life back to normal, cancer hit again. So since 2015, I haven't had any problems. So at the moment, I'm looking forward to doing my master's degree in computer games, art and design. And I'm not going to let this condition put me down. So I'm going to fight for it. Thank you.
So with this, we come to the end of our lectures for today. Most of us are going to be around, so by all means, catch up with us if you have any questions. And Lauren now is going to split you into 2 groups, 1 and 2. One to go and do the lab activity, and the other one to visit our exhibition, and then you will swap over in the middle of the slot. So thank you very much for your attention. And Lauren?
I think I'll just use my voice because it's loud enough. So I have emailed most teachers what their groups are, but just due to a few changes today and the schools not turning up, there has been a slight change. So this will be the group in